Hello guys, welcome to my channel. So as you know, we previously studied about the cell membrane. We are studying about the cell membrane, and so we studied about the phospholipids and glycolipids, how they are arranged and how they are manufactured. So now we'll study about the membrane proteins. As you know, the cell membrane has, beside the phospholipid, it has a membrane protein too. So how these membrane proteins are inserted in the cell membrane. So so all membrane proteins have a unique orientation in lipid bilayer, which is very essential for their function. So for example, for a transmembrane receptor protein, so the part of the protein that receives a signal from the outside of the cell, that is an outer environment of the cell, whereas the part that passes along the signal must be in the cytosol, because this transmembrane protein receives a signal. So it receives a signal from the outside, and it should, the another tail of the protein should be inside the cell, that is inside the cytosol. So this orientation is consequence of a way in which membrane proteins are synthesized. So for example, let's see. Right, so I will draw here. Right, this is the cell membrane. Right, so this is the cell membrane and the space I leave for the membrane protein. So how it will come here? Right, so first I will make here uh, a polypeptide chain. CH. All right, so this has negative charge, of course, and this will be positive. So like the chain goes on, and so it will be like this again. Negative hydrogen C. All right, so oops, sorry. So this is just an example of polypeptide chain. It is not straight though, but it would be like this. So that's just a simple example. So why I draw this polypeptide chain? The reason is the portion of the transmembrane protein located on either side of the lipid bilayer are connected by a specialized membrane spanning segment of polypeptide chain. So in between of this, so a part of membrane protein will be outside and the part will be inside, but in between there is a segment of polypeptide chain. Polypeptide chain. All right. So this segments run through the hydrophobic environment, because you know the tails are hydrophobic. So this segment runs through the hydrophobic environment of the interior of the lipid bilayer, are composed largely of amino acids. So this polypeptide chain is composed of amino acids with hydrophobic side chain. So this polypeptide will have an hydrophobic side chain, because these side chains cannot form favorable interaction with water molecules. So they prefer to interact with the hydrophobic tails of the lipid molecule where no water is present. But in contrast to hydrophobic side chain, in contrast to the hydrophobic side chains of the polypeptide chain, however, the peptide bonds, and this are the peptide bonds. Right, so these are the peptide bonds. All right, yeah. So in contrast to the hydrophobic side chain, however, the peptide bonds that join the successive amino acids, right? So this is joining the amino acids in a protein are normally polar, and because of this polar, the partial negative and the partial positive charges, The polypeptide backbone is also hydrophilic. As because the water is absent from the interior, there is no water present in between this part. There's no water present in this interior of the bilayer. Atoms forming the backbone, so this atom will form the backbone of the transmembrane protein, are driven to form hydrogen bonds with one another. Right, so what this polypeptide chain will do, it will form a helix structure in between this lipid bilayer. So we'll draw it here. Right, something like this. I'm not that much in drawing the diagrams on something like this, but just imagine this polypeptide chain will form a helix-like structure, alpha helix-like structure in between this lipid bilayer. So why it will form this structure? Because what will happen, the hydrogen bonds that are here and here present, so it will form hydrogen bonds that will make this hydrophilic as well. So the structure forming the helix, because water is absent from the interior of the bilayer, atoms forming the backbone are driven to form hydrogen bonds with one another. So it will form an hydrogen bonds with one another. All right, it will form an hydrogen bonds, and this hydrogen bond is maximized so this will be maximized when it will form an alpha helix, because if it's like straight like this, the hydrogen bond will not form that much, but if it forms a structure like an alpha helix, the hydrogen bonds will be maximized. And if the polypeptide chain forms a regular alpha helix, and so the great majority of the membrane spanning segment of polypeptide chain traverse the bilayer as alpha helices. So for the transmembrane protein, they will travel like this as an alpha helices. And in this membrane spanning alpha helices, the hydrophobic side, so what will happen? The hydrophobic side chain are exposed to the outside of the helix. So this will be in contact with, right? Sorry, I'm back. My marker was done. Right, so as I was saying, so the outside of the helix, the hydrophobic side of the helix will be in contact with the hydrophobic tails of the lipid bilayer. All right. So in many transmembrane proteins, the polypeptide chain crossing the membranes only once. So it will just cross once. But many of the single pass transmembrane proteins are receptors for extracellular signals. There are other transmembrane proteins as well that function as a channels. So like for the single one, it will be functioning as just for the extracellular signals to pass on inside the cell. But there are some other transmembrane proteins, they function as a channels, right? So forming an aqueous pore across the lipid bilayer to allow small and water soluble molecules to cross the membrane. So such channel cannot be formed by proteins with a single transmembrane alpha helix. So such channels will not be able to form if it's like this shape, just a single alpha helix. So what will happen in that situation? Instead of a single one, they will be usually consist of a series of helices that cross the bilayer a number of times. So in many of these multi-passes transmembrane proteins, one or more of the membrane spanning region are amphipathic, formed from alpha helices that contain both the hydrophobic and the hydrophilic amino acid side chains. So what will happen in the multiple pass? So like there will be multiple helices passing down on this side, and it will form like a cylindrical structure. I cannot draw 3D, but it would be something like this on three side. So these sides are actually done by the hydrophobic side chains, and they are in contact with the hydrophobic bilipid layer. But in the middle, so there will be like a middle. So there will be a middle aqueous pore, and there will be a channel to pass the water molecules from this transmembrane protein. So, and it will be a form of water-filled channel. All right. So on your screen, you will see a better picture for this. So this is how it will make the arrangement. All right, so the alpha helix is by far the most common form of the polypeptide chain that crosses a lipid bilayer. But the polypeptide chain of some of the transmembrane protein crosses the lipid bilayer as a beta sheet as well. So the alpha helix is the most common one. But there are some transmembrane protein where the polypeptide chain passes as a beta sheet or beta barrel, or a keg-like structures that is called a beta barrel. So, like as expected in this beta barrel as well, the amino acid side chain that faces the inside of the barrel and therefore line the aqueous channel and are mostly hydrophilic, while the outside of the barrel will be, of course, it will be hydrophobic, that will be lining with the hydrophobic tails of the lipid bilayer. Right, the most striking example of the beta barrel is porine protein. This protein is found in mitochondrial and the bacterial outer membrane, which form a large water-filled pores. So the mitochondria and some bacteria are surrounded by double membrane, and porins allow the passage of small nutrients and metabolites and inorganic ions across their outer membrane while preventing unwanted large molecules from crossing. Right, and you will see this how this beta barrel looks like. I cannot draw that picture, but you can see this image. This is how the beta barrel looks like. So this was the membrane proteins, and like just for the knowledge, like to understand the membrane proteins, what the biochemists do, like they have to separate the membrane protein from the cell membrane, and like as it is a partly aqueous and partly fatty acids, it's hard for them to do so. So how they did for the most membrane protein, the first step in the separation process involves solubilizing the membrane with the agent that destroyed the lipid bilayer by disrupting the hydrophobic association. So the most widely used disruptive agent are detergents. So they use detergent to separate this membrane proteins. So this the detergents are small amphipathic lipid-like molecules. It is same like lipid-like molecules, but as membrane phospholipids in that they only have a single hydrophobic chain, not double. So so a single hydrophobic tail, not a double tail, because they have as they have one tail, the detergent molecules are shaped like cones. So these are shaped like cylindrical as we studied, but the detergents are shaped like cones. And as they are shaped like cones, you remember like when they are inserted in water, they will form meshals rather than forming a bilayer as do the phospholipid. Right? When mixed in a great excess, so like when this detergent are mixed in a great excess with the membranes, what happens? The hydrophobic ends of the detergent molecule interact with the membrane spanning hydrophobic region of transmembrane protein as well as with the hydrophobic tails of the lipid bilayer of the phospholipid molecule, thereby disrupting the lipid bilayer and separating the proteins from the most of the phospholipid. And because the other end of the detergent molecule is hydrophilic, this interaction bring the membrane proteins into solution as a protein detergent complexes. So this membrane protein will be with the detergent and it will form a protein detergent complexes. At the same time, the detergent solubilizes the phospholipids. So the protein detergent complexes can then be separated from one another and from lipid detergent complexes for further analysis. So this is how it goes on for study. And just this was just for the knowledge or to know how they solubilize these things. Right. So the other than the transmembrane protein, there are protein that are located almost entirely in the cytosol and associated with the cytosolic half of the lipid bilayer by an amphipathic alpha helix exposed on the surface of the protein. You can see the picture on your screen. So that is number B. There is some proteins lie entirely outside the bilayer on one side of or the other, attached to the membrane only by one or more covalently attached lipid groups, that is C. And yet there are other proteins as well bound indirectly to one or the other face of the membrane the held in place only by their interaction with other membrane protein, and that is D. So this is how different kinds of protein. And so the protein that are like this are called integral membrane protein, but the remaining membrane proteins are just a peripheral membrane protein that they can be released from the membrane by more gentle extraction procedure. Not like for this, it will require us detergent to solubilize the phospholipids. Right. So now the cell membrane, if you know, the cell membrane is really thin, is extremely thin and fragile. So it would require actually nearly 10,000 cell membrane laid on top of one another to achieve a size of a thickness of the paper, right? So you can imagine how thin and fragile the cell membranes are. So what will happen? So the most cell membranes are strengthened and supported by a framework of protein attached to the transmembrane protein. So like for example, plant, yeast and bacteria, uh, the shell shape and the mechanical properties are conferred by the rigid cell wall that are made up of proteins and sugars and other macromolecules. But plasma membrane for an animal cell, that is for includes us as well, that stabilizes by a meshwork of fibrous proteins called cell cortex. So it is supported by fibrous protein called as cell cortex. All right. So the cortex of a human red blood cell is relatively very simple and regular structure. So that is why it was well studied. So these cells are small and have a distinctive flattened shape, as you know the shape of the RBC. So the main component of that cortex is, so the main component of the RBC, red blood cell cortex. So the main component of the red blood cell cortex is a dimeric protein, that is spectrin. All right, and the spectrin is a long, thin, and flexible rod, and that is about like 100 nanometers in length, and it forms a what it is. It forms a meshwork that provides support for the plasma membrane and maintains the cell biconcave shape. So that is why the shape of the red cell is biconcave. So the spectrin meshwork is connected to the membrane, the cell membrane, through the intracellular attachment proteins that links the spectrin to the specific transmembrane protein. So you can see on your screen. So this is a spectrin meshwork that forms the red cell cortex in the human red blood cell. So the spectrin, as you can see, are linked end to end to form the long tetramers, and the spectrin tetramers together with a small number of actin molecules are linked together in a mesh. So this mesh, this network is attached to the plasma membrane by binding of at least two types of attachment proteins and or the two kinds of the transmembrane protein. So you can see that in yellow and blue, that is an attachment protein, and the green and brown is a transmembrane protein. So this is how the spectrum supports the cell membrane. Great. So as we know, the lipid bilayer is two-dimensional. So because of this two-dimensional structure, two-dimensional fluid structure, we can say that it can freely move. So as a lipid can freely move, the proteins in this lipid bilayer can also make a lateral diffusion. So the scientists did a test. What they did, they experimented fusing a mouse cell with a human cell to form a double-sized hybrid cell, and then monitored the distribution of certain house and human plasma membrane proteins. So what happened at the first, the mouse and the human proteins are confined at their two own halves. But as the time goes, but within like in half an hour, so the two sets of proteins formed hybrids become, so the two sets of proteins become evenly mixed over the entire cell. So they come together and they just mix, and it becomes just like a one cell. So what does that mean? But the cells have their own way of confining particular protein to localized areas. within the bilayer membrane, thereby creating functionally specialized region or membrane domains on the cell or the organelle surfaces. So the plasma membrane is, the plasma membrane protein can be tethered to the structure outside of the cell. So for example, if there is a structure outside of the cell, so it will, the membrane protein, if this protein is important and it should be localized, so it will be attached to this outside of the cell. So and the extracellular matrix or adjacent the cell, or to relatively immobile structure inside the cell, especially to the cell cortex. So like where there's a cell cortex, it's not moving, so the protein might be fixed with it. So additionally, cell can create barriers as well that restrict particular membrane components to one membrane domain only. As for example, in epithelial cell that lines our gut. For example, it is really important that transport proteins, the transport protein involved in the uptake of nutrients from the gut, be confined to the apical surface of the cells. So the surface that faces the gut contents, right? And that the other transport proteins involved in the export of solutes out of the epithelial cells into the tissues and the bloodstream be confined to the basal and the lateral surfaces only. So suppose if there is like over here, suppose this is the this is the apical surface, this is the lateral, and this is the basal surface. So there is the protein on here, here, and here. So it won't be reached, like this protein will go to the top, right? And so it would be actually like this. So the cells and the tissues. All right. So what this will do? So this membrane proteins are uh, asymmetrically distributed in the cell, right? In the gut cell. So what? Right. So the asymmetry is maintained by a barrier formed along the line where the cell is sealed to the adjacent epithelial cells. That is called a tight junction. So there will be a junction here, so that will not allow this to move up on the on that side. So at this side, the specialized junctional protein form a continuous belt around the cell where the cell contracts its neighbor, creating a seal between an adjacent plasma membranes. Membrane proteins cannot diffuse and pass the junctions. Right, and one more thing for the cell membrane. The cell membrane surface or the cell surface is coated with carbohydrates. As we know that the plasma membrane have sugar covalently attached to them, that is glycolipids. So, but the protein of the plasma membrane also is attached with the sugar. So, the great majority of proteins have short chains of sugar, that is called oligosaccharides, linked to them, and they are called glycoproteins. If the sugar is attached to this protein, it will be called glycoproteins. Other membrane proteins, also known as the proteoglycans, contain one or more long polysaccharide chains. All of the carbohydrates on the glycoproteins or the proteoglycans and the glycolipids is located on the outside of the plasma membrane, where it forms a sugar coating called the carbohydrate layer or glycocalyx. Calyx. All right, so this all will have a sugar chain attached to this. All right, so this layer of carbohydrates, what this layer will do, helps protect the cell surface from the mechanical damages. As oligosaccharide and polysaccharides absorb water, they also give a cell slimy surface, which help the motile cells such as white blood cells to squeeze through the narrow spaces and prevent blood cells from sticking to one another or the walls of the blood vessel. And you can see in this image, this is how this sugars are attached to the cell surface. So the oligosaccharide side chain attached to the membrane, glycolipids and the glycoproteins. All right. So the cell surface carbohydrates do more than just protect and lubricate the cell. So the functions are more for the cell surface carbohydrates. So what they do? So the important role is for the cell carbohydrate is in the cell to cell recognition. Cell to cell recognition. So Just as many protein will recognize a particular site of the another protein. Like for example, proteins called lectins are specialized to bind to a particular oligosaccharide side chain. So the oligosaccharide side chains of glycoprotein and the glycolipids, although short, so like it would be like normally, like as we studied before when we studied sugars, so the oligosaccharide is usually less than 15 or 10 sugar units, and are but they are enormously diverse. Like unlike proteins, the amino acids are all joined together in a linear chain by identical peptide bonds. Sugar does not have that case. Sugar can be joined in many different arrangements, often forming elaborate branch structure, as we discussed further before in the video when we discussed about the sugar. So that's why this oligosaccharides or the polysaccharides are really hard to study because they have a complex or large branches. So using a variety of covalent linkages, even three different sugars can form hundreds of different trisaccharides. So this is how it complex it is. So the carbohydrate layer on the surface of the cell in a multicellular organism, so what this will do, it will be serving as a kind of distinctive clothing, different types of clothing of the cell, like a police officer uniform that is a characteristics of each cell type and is recognized by other cell types that interact with it. So all this the cell carbohydrate layer of the cell membrane or the cells will be as a clothing. They have a different clothing, and it will be like a police officer or different kinds, and they will know this cloth has this, oh, this is this guy, I need to talk with this guy, like it's something like that. All right, and so it is the characteristics of each cell type and is recognized by the other cell type and that is interact with it. So the specific oligosaccharide in carbohydrate layer are involved. For example, in the recognition of an egg by a sperm, right? So there is a specific oligosaccharide in the cell of the sperm and the egg. So they have special things, so they will recognize each other, and then this is how it meets. So similarly, in the early stage of bacterial infection, so the carbohydrate on the surface of a white blood cell. So, for example, this is endothelial cell lining, right? That are made by the endothelial cell. So this endothelial cell will make a transmembrane protein called lactin. I will put it here. Lactin, right? That will be here. So this lactin is recognized by a okay. So this endothelial cell. So for example, the blood vessel of the endothelial cell. So if this is a site of infection, so what this blood vessel or the endothelial cell will do, it will generate a transmembrane protein that is lactin, or specialized transmembrane protein you can call, that is known as lactin, and this lactin is recognized by the neutrophil, that is the white blood cell, that is the first line of defense of our body. Neutrophil, the carbohydrate, that is the carbohydrate surface. So for example, this is neutrophil. I'll put an N for the neutrophil, and this neutrophil, that is the carbohydrates on this neutrophil, are specialized to recognize the protein that is lactin. So this oligosaccharide, so for example, this is the oligosaccharide of neutrophil. So this will recognize this, and it will be attached to this, and there is a process, and after a process, this white blood cell or the neutrophil will migrate to the infected tissue where they will help to destroy the invading bacteria. So the cell surface carbohydrate is really important, not only to protect and lubricate the cell, but even to the cell to cell recognition that will help this kind of functions. Alright guys, this was a very detailed topic of cell membrane. So we covered how the membrane are manufactured, how they are, what are their functions, the proteins, lipids, and on the last, the cell surface carbohydrates. Alright guys, thank you guys for watching, and please don't forget to subscribe my channel.